All right, hi guys. So these notes are going to be the Mendelian and non-Mendelian notes. I'm not going to cover the Mendel part because we covered that previously. So this is just going to be a little bit of a review and then get more into the non-Mendelian parts of the notes because I know we didn't get all the way through those in class. So if you feel pretty comfortable with Punnett squares, you can go ahead and skip through the first couple of minutes of the video and then get to the part where we didn't cover it in class. This is about slide 9 of your PowerPoint presentation on the Mendelian and non-Mendelian notes. So here we go. Punnett squares. I just want to remind you guys when you're setting them up, we got one parent here, we got one parent here. This is the parental genotype, the big A, so they'd be considered homozygous dominant. This is parent number 2. This is their genotype. It would be homozygous recessive. And just remember, because of the laws of independent assortment and the law of segregation, you're going to list one of the possible alleles that the parent can pass on above this square and one over here, because each one of these represents a gamete or a sperm that can be passed on. And each one of these represents a version for this one mono trait, okay, one trait. Um, one gene, it represents an egg that could be passed on, a variation of that egg. And so that means that each of these squares that you see inside the Punnett square is going to be what happens after sperm meets egg or fertilization. You end up, it represents the genotype of a diploid individual. And so that's where you're going to get this diploid individual that is the big A from parent 1, the big allele from parent 1, that one version, and then the small allele or the recessive allele from parent number 2. And they're going to be what we call heterozygous, or what Mendel called is a hybrid. Okay. And so it's 1 out of 4 because there are 4 possibilities. And so if this were, little a, and little a, so you had a homozygous recessive parent with a homozygous recessive parent, 100% of them will be homozygous recessive. If you had the homozygous dominant with the homozygous recessive, 100% of them would be heterozygous. But each one of these represents a possibility that this sperm could fertilize this egg, this sperm could fertilize that same egg. Okay, so it's all the possible combinations, 1 out of 4 probability, or 25% for each one of those. Okay. All right. I think this is the one that had the typo where it says 2 heterozygous parents, and these are definitely not 2 heterozygous parents. So what I'm going to do is, and I think we corrected this one in class, we either redid it with 2 heterozygous parents, or we did it with, you could do 1 heterozygous parent and 1 homozygous recessive. Okay, and that's what's actually depicted in the Punnett square as you see it right now. Is you have a heterozygous parent right here, you have a homozygous recessive parent right here. The only allele that a homozygous recessive parent can pass on is the little t. That's the only one they've got. And then a heterozygous parent can pass on either the big T or the little t. They're going to assort independently from each other. And so what you're going to end up with is this combination of all the possible offspring. So you could get 25% with, that's not correct. What you could get is you could end up with 50% as heterozygous, 50% as heterozygous, and then 50% as homozygous recessive. So these genotypes are actually for 2 heterozygous parents being crossed, which is not what's pictured. So what you should notice though is this is an example from before. So if I kind of ignored this part that I wrote in, okay, of the other depiction, 2 heterozygous parents, or what Mendel called a hybrid cross, monohybrid cross, you're going to get a 1 to 2 to 1 ratio of the genotype. If you remember, those, the genotypic ratio for a monohybrid cross in Mendelian genetics should be 1 to 2 to 1. That's a ratio you need to know. And then the phenotypic ratio for a monohybrid cross in Mendelian genetics is 3 to 1. So 3 will appear to have the dominant trait, and 1 will appear to have the recessive trait. And remember, only in the recessive individual does their phenotype match their genotype if they are homozygous recessive. That's the only one we know for sure because of those three that are tall, that 75% that are tall, we don't know whether that's going to be the homozygous tall or whether that's going to be a heterozygous. Okay, so the only genotype that perfectly matches the phenotype is if they are recessive for both versions of the alleles. And remember that recessive does not mean it occurs less often in the population. Remember the fact that you all have 5 fingers and 5 toes on each one of your hands, your feet, your digits, and that is a recessive trait. Okay, so it does not mean that it occurs less often in the population. It just means it is a non-functioning version of that gene, or it is recessive and or masked by the other functional version of that gene. All right, the non-Mendelian traits. So here we go. They can be controlled by more than one gene. They can be controlled by multiple alleles. Blood typing has 3 alleles. They can be controlled by multiple alleles and more than one gene. You can have not fully dominant, not fully recessive. So you can have what we call codominance, incomplete dominance. Okay, so these are all the patterns of inheritance that do not follow Mendel's laws, and there are quite a few of them. And what we find more often than not is as we explore human genetics and we get to know more and more and more and more and more about humans, is that fewer and fewer and fewer of our traits are actually due to Mendelian genetics. Most of them are due to what we call modern genetics. Okay, non-Mendelian genetic example number 1, blood typing. So here we go. Blood typing actually accounts for 2 types of modern genetics. They are an example of what we call multiple alleles. Okay, the idea that you can have the A allele, okay, the little caret mark, remember, is a superscript. I just can't do superscripts on Zoom. And then I superscript B, the B allele. And then you can also have a recessive version of the allele, which is depicted by the lowercase I. So what that means is that if you have the allele IA, you make proteins, blood proteins on your red blood cells, your hemoglobin, that's protein A. You make a different protein if you have IB. Okay, so there's an A version of the surface protein and there's a B version of the surface protein. If you are recessive, you do not make that surface protein. Hence, a recessive trait is something that isn't a functioning version of the gene, which means you don't make the surface protein for your red blood cell, A or B. You just don't make it. So you are lowercase I. Now, when we talk about blood types, you can be type A. All right, you can be homozygous dominant, IA, or you can be heterozygous, IA, and then lowercase I. The lowercase I won't make a functioning version, but this allele right here will. And so you'll have enough of those to have type A blood. Same thing for type B. And then I'm going to skip over here to type O. This is a typo on type O. Sorry for the pun right there. So these are incorrect. The only way to have blood type O is to be lowercase I. You have 2 non-functioning versions of the gene for the surface protein on your red blood cells. You lack all surface proteins. Rhesus factors are a totally different thing. That's the positives and the negatives, and we'll talk about that later. But we're just talking about type A, type B, and type O. So you do have 3 different alleles that can code for the different blood types. And then I'm going to talk about type AB, because AB is actually an example of, I'm going to type it down here, what we call codominance. Okay. That's where both the version A and version B of the alleles will be expressed together as if they are both dominant. So someone who has type AB blood, their red blood cells contain both the A surface protein and the B surface protein. Okay, so their red blood cells have both. And we did talk about in class how that makes blood type AB. If you're AB positive, you have all the surface proteins that you could possibly have, which makes you the universal recipient. You can get anybody's blood type donated to you, and it doesn't matter. You can accept all of them because you have all the surface proteins. If you are type O negative, you lack all surface proteins, which means everybody really likes your blood. They're not going to reject it. So you are the universal donor. But as far as genetics goes, Blood typing is an example of both multiple alleles for A, B, and I, lowercase I, and then codominance for the fact that both type A and type B are expressed together. Okay, so if you are heterozygous IA and and you have the IB, that's blood type AB. Okay, so a different version is called polygenic, and this is one where you can have three different genes, four different genes, five different genes that all control for the same phenotypic trait or physical expression of your genetics. So, skin color, eye color, hair color, height, seed color in plants, fur color in several other organisms are all going to be due to what we call polygenics, polygenic inheritance, or it's a polygenic trait. It just means it's controlled by more than one gene. And for the most part, it's controlled by the presence of a functioning version of each of those genes. Therefore, if we're doing seed coat color, if you are homozygous dominant for all three genes, so big A, big B, and big C, so dominant for all three of those, then you are going to have a dark seed coat color. If you are recessive for those three genes, little a, little b, little c, you are going to be really pale white. You are lacking the functioning genes or the functioning allele to produce the pigment, because if you are recessive, you are a non-functioning version of that gene. Therefore, you make no coat color pigment for that wheat. And so it actually does work pretty well along a sliding, almost like a bell curve with polygenic inheritance. If you have a seed that is heterozygous for all three, and you cross it with a heterozygous for all three, here are the chances. The majority, 20 out of 64, are going to look just like the parental, but you could have a 1 out of 64 chance of having a homozygous recessive for all three traits, or having homozygous dominant for all three traits. Okay, so it is kind of this sliding scale, just purely based on the presence of how many dominant alleles you have. Okay, so this is another one kind of like codominance. So incomplete dominance is going to be with these snapdragons right here. So you have a red snapdragon, and let's say that the genotype is going to be big R, big R. You have a recessive snapdragon, and so white snapdragon is homozygous recessive, so let's say that's little r, little r. You cross those two together, and all of the genotypes of these offspring should be big R, little r. Should be 100% heterozygote, okay? If they are incomplete dominant genes, if it's incomplete dominance or incomplete inheritance, what that means is you have a blending of the two parental phenotypes in the expression of the offspring. Because this snapdragon only contains one dominant allele or one functioning version, it makes enough of the red pigment to partially express it, but not enough to be red. And so what ends up happening is the offspring end up being a blending of the two parental phenotypes. And so instead of getting a red snapdragon and a white snapdragon, right? Because if this were Mendelian inheritance, you would expect 3 and 1. Actually, you'd expect 100% red. Sorry, you'd expect 100% red if this is Mendelian inheritance. But because it's incomplete dominance, you get pink, because pink is a blending between red and white. Okay, so incomplete dominance means that the dominant allele does not completely mask the presence of the recessive, and you end up with a blending of the two. This does happen in snapdragons. In tulips, however, it's codominance. And so what would happen is you would end up with both of them being expressed together. And so when that happens, you end up with red and white striped offspring. So that's the difference between seeing codominance versus seeing incomplete dominance. Incomplete dominance is a blending of the two, red and white make pink. Codominance is an expression of both. So red and white make red and white striped. Okay. So again, this is like perfect example of the red and white stripe. They're both appearing together. Codominance is very similar. It's just they're both going to be expressed with equally without blending. You've got a roan cow, which is a special color for red, and you've got a white cow, and you blend the two together, and you're going to get red and white spotting. It's the coat color that they have. Again, AB, blood typing AB is the heterozygote of both the A allele and the B allele, and you express them both together without blending. Okay, so I apologize for our colorblind students. You may not be able to tell. There's a 12 here, there's a 6 here, and I don't think there's a number here. I can't see anything. But certain traits are coded for on your sex chromosomes. We say that those are sex linked. If you are female, you have two X chromosomes, and if you are male, you have one X chromosome and a Y chromosome. Most of the time, when you come across something that is what we call X linked, that means it's on the X chromosome. I just want you to be aware that the phrase sex linked does not mean it has to be on the X chromosome. It could be a gene on the Y chromosome. Usually they'll say specifically on the AP an X linked trait. Probably you can assume sex linked also means X linked, unless they say otherwise, because there are very few genes besides SRY genes on the Y chromosome. And those are just for coding for the fact that you are male. So testosterone production and then secondary sex characteristics once puberty happens. This is just an example. Color blindness, red green color blindness is what we call an X linked trait. It is on the X chromosome, which means to make you biologically male, you get the Y from your father. So if you are red green colorblind, you did inherit that from the X on your mother, unless you happen to have an B Klinefelters. So a non-disjunction happened and one of those X's from your dad stuck together. So you're XXY. We did talk about a little bit in class how females, they are XX, which means you are redundant. So if one of your copies of your X chromosome has a genetic trait on it that is unfavorable, but the other one has a normal functioning version of that trait, you will be what's considered a carrier for that disorder, and you can pass that on to your offspring. But you may have a normal enough functioning life that you don't have the disorder yourself. So example of that, like in the royal family in Great Britain, would be hemophilia. Queen Victoria was a carrier for hemophilia and passed that on to a lot of her offspring. But she had a normal functioning life because of her redundant X copy. And that's something we did talk about, because when you look at family trees, if it is an X-linked trait, it will show up in males more than females, because the males don't have the protection of that redundant X, or the benefit of having possibly a normal functioning version of the X gene. Okay, these are going to be pedigrees. If you do like AKC registration or thoroughbred racing, you might have seen these. Any type of show animal, you might have seen a pedigree before. It's basically a family tree. It's a graphic representation of a family tree. For people, so we say that the squares are males, the circles are females. If they are colored in, they have the disorder or the trait that you're looking for. If they are not colored in, they do not have the trait you are looking for. Most of the time when they give you a pedigree on the AP, and they do like to, either in FRQ or multiple choice form, they will give you a key for it that tells you like affected, unaffected. But it is assumed that you know male and female. If there is a line that is appearing between the two, that means it is a mating and they produced offspring. These are the offspring. Each generation is listed on a different line. So this is the parental generation. This is the filial generation, one of the kids. And then if these individuals had offspring, so if this individual had, got to get a circle, a mating with this individual, their offspring would then be on the next line down. Each generation is on the same line, okay? So this male offspring, it has the trait. This female offspring does not have the trait. So if you have two parents that are both affected, But one child that is not affected, I would say that this is a dominant trait, but both of these parents are heterozygous. So if you were to look at it, okay, so if this person's heterozygous and they mate with this person who's heterozygous, remembering your Punnett squares, then if this were Mendelian genetics, you could say that there's a 75% chance that they're going to be affected and a 25% chance that they're not going to be affected. And so this male is affected, but you don't know whether they got the big R from one parent and another big R from a parent, or whether they got the big R from a parent and a little r from a parent. You don't know. So you can't really tell the genotype. If you can't tell the second letter, you put a line. So what that represents is that they are big R, or they are big R, little r. You don't know what it is, so you just put a line down. However, this individual that is unaffected, I know for certain that they got little r, because if they had received either one of these dominant alleles from either parent, they would have also expressed the trait. Okay. All right, so these are just some examples of the pedigrees that we're looking at. Autosomal dominant traits, they're usually going to appear, you're going to see them in every generation. You're going to see them pretty equally in males and females. And remember, autosomal means anything but your sex chromosomes. So this is going to be any disorder or trait, could just be a trait, that's listed on chromosomes 1 through 22. Your 23rd pair is either your XX or your XY, and those are your sex chromosomes. So autosomal is referring to anything except those. So this would not be for an X-linked trait. Autosomal dominant traits, you're going to see it pretty typically. One parent has to be affected in order to have affected children, but not all the children have to be affected. So they did have an unaffected son and an unaffected daughter, which tells me that mom here is heterozygous. So if this is autosomal dominant, that means that you can, let's just say H, so homozygous dominant, or you can be heterozygous and you will express it, which means if you have a child that has 2 recessive alleles, okay, little h, and another child that has 2 recessive alleles, little h, right? In order to have that, because of the laws of independent assortment, both parents have, because you get one allele from one parent and one allele from the other, both parents have to have the recessive allele present. So this unaffected male is homozygous recessive, and this affected female is heterozygous. That is how this mating is able to produce offspring that are affected and offspring that are unaffected. Okay, autosomal recessive is going to appear less frequently, and really where it's going to pop up is if you see 2 parents unaffected that have a child who is affected. Woo, go back. Oh my goodness. Okay. What is happening here is that both of these parents must be heterozygous. Okay. In order to have an affected child. Because if they are heterozygous, that means that dominant normal functioning allele allows them to not be affected by this recessive trait or disease, but that they do have a child who is affected means that they've produced a child that is homozygous recessive. In order to produce a child that is homozygous recessive, they must both have that recessive allele available to pass on in their egg and sperm, which means that because they are unaffected, but they have to have that recessive allele, they both have to be heterozygous, okay? One of these could be like, you could have a heterozygous parent and a homozygous recessive parent, but then they would be colored in red. Because this mating also had a child who is affected, and they would be little h, that meant that they got the little h from one parent. They had to get a second copy of the recessive allele another little H from their other parent. And so this female right here is heterozygous as well. Okay. This is where the heterozygotes play an interesting dynamic in keeping autosomal recessive diseases alive. Because most of the time people would think, oh, well, if it's a recessive trait, eventually it'll just be dominated over and it'll die out of the population, especially if it's lethal or something dangerous. And so eventually it'll just go away. And it doesn't, because heterozygotes coming together and having multiple offspring, eventually it'll just keep popping back up in the population. And that's where you kind of have this resiliency of these autosomal recessive traits, is because there are so many people out there that are heterozygous. And it doesn't necessarily have to be a bad thing. It can be good to be a heterozygote. Oh no, my pedigree is in the way. All right, well, we'll talk about it. If it is recessive X-linked, it's going to appear more in males. That's because they only have one copy. They do not have the redundant copy. What I mean by redundant copy is, please remember when we talked way back when with meiosis and egg formation, that one of your X chromosomes, if you are genetically female and you are XX, one of your X chromosomes deactivates because it's too many active copy, too many genes in your cells, and one of them super condenses. So if you remember, through methylation and acetylation, it's going to deacetylation, it's going to super condense and coil up to where you can't copy any of the genes in order to make proteins. And so that is essentially turned off. And that's going to be one of your X copies, and it's random which one it is. And we call that turned off X copy a polar body. X chromosome. And so it makes every female kind of a mosaic patchwork of which X is activated and which one is deactivated. But that provides you with a redundancy of having functional version of your X chromosome. Males, however, do not have that benefit because they are XY. They will express everything they have that's on their X chromosome, and that they inherited from the egg, because that can only pass on XX. If they receive the Y chromosome, that's from the sperm, because sperm determines the sex of the offspring, because you can have X or you can have Y sperm that are passed on. And so males are the ones that determine the sex of the offspring. If you are in that redundancy, okay, if you don't have two normal functioning versions of the X chromosome, one is, let's say, dominant and one's recessive in a recessive X-linked disease, you then, in that, being turned on, turned off kind of thing, right? That's in your autosomal cells for females. That's in your body cells. So you aren't affected. However, remember, when you undergo meiosis and you go to make eggs, that you can then, pass on, that's the genes in meiosis, the eggs that you pass on to your offspring, both X's are activated and randomly assort. And so you can pass on, even though you're normal functioning enough to not really have the disorder, you can pass on either a functioning version of the X, that gene on the X chromosome, or you can pass on a non-functioning version of that gene on the X chromosome. So just because you don't express the disorder in yourself, in your autosomal cells, doesn't mean that you can't pass it to your offspring, because in meiosis, both of them are activated when you're doing egg formation. And because of random assortment and independent assortment, they're going to, you can pass on both of them in either one of the versions that you make, in the eggs that you make. So in a pedigree, you are considered a carrier, which is why you have this line through it. This kind of half color is you're half affected, half not affected. Sometimes in a pedigree on the AP, they will give this to you. And sometimes they will give you in the paragraph some genotypes in here, and they'll expect you to recognize that some of them are carriers, and they will not give it to you filled in. So you can see either one on the AP. Okay. These right here, if you are a carrier, you are a heterozygote. Please remember that we always, if it is X linked, remember again, the caret represents the superscript, okay? So it almost looks like an exponent. It's X with whatever it is, so we'll do big H, so we'll act like it's hemophilia, and then we'll do an X with the superscript of little h. So in case you don't know what like you would be like this, okay? If you were a carrier, it would look like this. If it is written on the X like this, it is an X-linked disorder, okay? All right, genetic diseases. So there are 3 types of single gene mutation disorders, and that's autosomal dominant, autosomal recessive, and X-linked. We talked about those. Some examples of those are Huntington's. Huntington's, unfortunately, is autosomal dominant. And really, you would think if it's a dominant disorder and it's lethal, and it is, it would die out of the population, except for the fact that when people pop up with Huntington's, it's not until later in life. So they can be as early as 30, but they can be as late as 40s, 50s, 60s that they pop that they start to display symptoms of Huntington's disease. It is lethal once you're diagnosed with Huntington's. There is no cure for it. It is an extremely shortened lifespan, and you will lose neurological control of your faculties with Huntington's and eventually pass, usually pretty quickly after diagnosis. And unfortunately, what that means is by the time you're diagnosed, you probably already have children. And so you probably already have offspring. And so then again, it gets passed in the population because it is autosomal dominant. So there's a pretty high chance that your children also have Huntington's. They do, however, have genetic testing for it. So if you do have a parent that ends up being diagnosed with Huntington's, you can get genetic testing that will tell you whether or not you have the potential of developing Huntington's. Duchenne's muscular dystrophy is X-linked recessive. So you do have to have, this Duchenne's will appear more often in males, inheriting the recessive X from a mother who is a carrier. And again, so this is an example of an X-linked trait that is, there are lots of therapies that you can live much longer now with muscular dystrophy. It is not the same death sentence that it used to be, but it is not a normal life. It's modified heavily with medications and therapies. Sickle cell anemia is actually one of those where we're going to talk about it a little bit more, and I like to talk about this one. So sickle cell anemia, it's different than you have sickle cell disease. So if you have sickle cell disease, you have the majority of your blood cells are sickle cell. And growing up, you can't run a lot because when you run, you're going to pump your blood. That means more blood circulating through your system really, really fast. All of your blood vessels are circular, which hence is why your red blood cells are circular, little kind of indented, almost like donut, like raft looking shapes. But if you have sickle cell disease, you have a mutation to where instead of your proteins forming this nice circular attraction of the 4 subunits that make up your hemoglobin or your red blood cells, you have one where they kind of twist and they actually repel each other. So instead of being hydrophilic, they're instead of being hydrophilic, one is hydrophobic, one is hydrophilic, and they repel each other. And so what you end up with is these crescent moon or sickle shaped cells. So you can kind of see like this is a really bad one, but this is, like this, right? The problem with these is that they also are much stickier and harder. They're more rigid and they don't carry oxygen. Okay. So if you have sickle cell disease and you go running, you're going to then circulate your blood faster, which means you're going to circulate all of those sickle shaped cells more, which means they have a higher chance of going through those round blood cells and blood vessels and getting stuck. And one will get stuck and then the other one will get stuck on it and they'll get stuck kind of in this chain of little crescent shape, crescent shape, crescent shape, and they form a blockage or a clot, which is why people who have sickle cell disease oftentimes have to have blood transfusions on a regular basis. They have a modified life. They have breathing problems. They don't exercise. They don't run around. And a lot of therapies. And of course, there's now CRISPR and gene modifications for people who have sickle cell. But it also forms an evolutionary advantage because if you have sickle cell anemia, What that means is you have enough normal functioning red blood cells to have a normal life. You have enough sickle shaped red blood cells that you appear to have anemia. And anemia is just a lack of iron. And it presents in the fact that you're usually a little tired because the iron in your blood, the heme group, is what holds on to the oxygen that then goes around and feeds your brain, your muscles, and all of your organs and your systems. So if you have enough normal functioning ones and enough non normal functioning ones, you're like, well, why would that be a good thing? Well, that's a good thing if you live in a malarial region. Malaria is a little tiny like parasite. Okay. So it's an endoparasite. It lives inside you. It gets inside you through mosquito bites. So mosquitoes are the vectors. So they'll bite a person who has malaria. The malarial worm will get inside them. They'll then bite you. And in an attempt to inject their numbing agent and suck more blood out of you, they will inject the worm, which will get into your circulatory system. And they like to hitch a ride on your red blood cells and make their way to your liver. They can't, however, hitch a ride on the sickle shaped red blood cells. They're too hard, they're too tough or rigid for them. They can't get in. Okay. So it actually doesn't make you immune, but it makes you what we call resistant. You're less likely to develop a full blown malarial infection if you have sickle cell anemia. And because you have some functioning red blood cells and some not, you are heterozygote. And so that's an example of what we call a heterozygote advantage. That's where if you are homozygous recessive, okay, autosomal recessive, if you're homozygous recessive to non functioning copies, you will have a shortened life, okay? And you will not live long with full blown sickle cell disease without lots of therapies and interventions. If you are dominant for normal functioning, then you don't have either of those. You produce nice, round, little raft looking, squishy, non sticky red blood cells. But if you are heterozygous, you have a resistance to malaria. So this is an example where a disorder, a disease in people has evolved as a defense against another disease or against a parasite. So this is where a disease and a parasite kind of evolve together because you will find more sickle cell anemic and sickle cell disease people genetically in regions that carry malaria. So in Africa, sub-Saharan Africa, Southeast Asia, India, malarial zones. It's just really interesting to see because as global temperatures start to rise, the malarial mosquitoes, there's very specific mosquitoes that can carry malaria, they're able to live in more and more regions. And so now, like malarial mosquitoes can be found in the Everglades in Florida. Cool stuff. Okay. So a person only needs one mutation of that gene in order to be affected if it's autosomal dominant. Because if you could have autosomal, excuse me, you could have homozygous dominant, but if you had homozygous dominant individual in a genetic disease that is autosomal dominant, both of your parents were also affected. And that's probably not going to happen. But you only need one copy. If it's autosomal dominant, you have one dominant copy and you will express it. So it will dominate over law of dominance. It will dominate over that recessive copy. Huntington's disease is an example of autosomal dominant. We talked about that just real briefly. It's the degenerative disorder. It basically is the death of your brain cells. And you lose your coordination, you lose your muscle movement, your inability to talk, inability to swallow, and eventually your inability to breathe because your diaphragm is a muscle as well. And you'll lose the coordination in that and be unable to breathe on your own. And it's pretty quick. It's only a couple of years after diagnosis. Marfan syndrome is the connective tissue that it causes. Affected individuals will be tall, thin, and have long limb fingers that you'll see, like long limbs and fingers. I think it was an NBA player that was actually diagnosed with Marfan syndrome. Um, the problem is that because of this disease with your connective tissue, they will be tall, they will be thin, they will be long. You know, they'll look like NBA players basically. But eventually there will be degradation of this connective tissue and it can lead to a lot of other issues. Oh, there's a box where there shouldn't be a box. Genetic diseases. So, in order to have a recessive, autosomal recessive genetic disease, a person needs two mutated copies. So, most of the time, the examples that I have would be like cystic fibrosis is one of them. Um, this would be one where both parents have to be carriers. You usually won't find an example where one parent is affected and one parent is heterozygous or a carrier, because most of the time, for most of these autosomal recessive disorders, especially when we're talking about diseases like cystic fibrosis, the person who has it won't live long enough to have offspring. Um, so there are more therapies with a cystic fibrosis individual. You can kind of see the lungs of a cystic fibrosis patient are cloudy. What's happening is that the mucus that they make that's normally inside your lungs, in case you didn't know, a membrane has to be moist or wet in order for gases exchange to happen across it. So your lungs do have to be wet in order for you to have oxygen and carbon dioxide exchange. And that's normal. Unfortunately, that also means that you have to produce a lot of mucus to keep them that way, but normally your mucus is thin. And it's just, you know, it's put out, it's secreted, it's absorbed, it's secreted, it's absorbed. It's a normal functioning thing. If, however, you have cystic fibrosis, your mucus is really, really thick. And you can't actually just reabsorb it because you also have a defect in your chloride ion channels in your tissues. And so you will not reabsorb the fluid that's in your lungs. So you have to go on medication therapies. And then back in the day, long ago, both of my grandparents happened to be carriers for cystic fibrosis. And so they ended up having to adopt. My father's adopted. But they had three biological children before their first was diagnosed with cystic fibrosis. And ultimately, all three of their children were diagnosed with cystic fibrosis. But back in the day, you'd have to put your child on basically like this ramp-like thing, where it's almost like a wedge, like this, okay? And your head would be down here, so you're downhill, okay? And this would be like your back region, and you're, you know, an adult, a parent would basically like pound on your back from the outside, not like hurt them, but enough that you could physically jar the fluid in their lungs loose. That was a thing. And especially if your child just was having so much like a hard time breathing. But it was a long time ago a much larger death sentence than it is today. Again, people with cystic fibrosis are living a lot longer. We have gene therapies, we have drug therapies. But it used to be when diagnosed with cystic fibrosis, and usually pretty early, you would be diagnosed with it. You unfortunately passed before the age of probably 11, 12, 13. So it's a much, 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 much shorter lifespan. Yeah. So both parents for cystic fibrosis, both parents have to be carriers for it. They both have to contain that recessive allele. And then again, sickle cell anemia, we just talked about that. There can be a heterozygote advantage. There can be an advantage to being a carrier for sickle cell disease, and that is that you have that resistance to malaria. So it's not always this like doom and gloom, horrible death sentence. It's just usually that's what we study because that's what people are interested in. Non-living factors that are responsible for influencing inherited traits is the idea of the sun. You can tan. So you can be born with, let's say you're homozygous recessive for all of the 5 alleles that are going to code for your skin color. So you have no dominant copies whatsoever. You are pale, pasty, alabaster white. But if you happen to live, let's say in Hawaii, and you wear sunscreen sometimes because you just don't want to fry like a lobster, but over time you will tan. Now, that tanning is just the idea that your cells in your skin is attempting to protect itself from the UV radiation that is permeating through all of the membranes and straight to your DNA where it can mutate it. And so it creates that pigment, that skin pigmentation as natural barrier to block the UV radiation. It doesn't block all of it, but it definitely blocks a lot of it. So wear your sunscreen. That also means that people who have darker pigmented skin naturally, they do have more of that sunscreen, that natural sunscreen. However, that does not mean they should not wear sunscreen. Everybody should wear sunscreen, protect you from skin cancer, because it's a little bit better to have some like vitamin D fortified orange juice and milk than it is to have skin cancer. So yes, if you wear sunscreen all the time, you're inside all the time, you will have lower vitamin D levels because your skin naturally makes that with the use of UV light. But that's better than skin cancer. So temperature influence in Siamese cats is kind of cool. Their cooler body regions, which are going to be their hands, their forepaws and their rear paws are going to be, and their head and feet are going to be darker, because those are cooler. And then where they're nice and warm is actually where it's lighter colored, the pigmentation under their skin. And then these are one of my favorite flowers, hydrangea. And this is going to be where they're blue when the pH is low. So we're talking like 1, 2, so more acidic pH. And they're pink when the soil is more basic. And you can actually do this naturally. You can buy supplements at the store. You can just go put coffee grounds on them, and you can change the pH. You can buy like Home Depot has like pH soil monitors for, I think it's like $2. And you can just amend the soil with like eggshells and coffee grounds and different things to make it more basic or more acidic. Bonefish meal, and then you can actually change the color of your hydrangea. It's really cool. They don't change instantly. It's not that. Like they'll change across a growing season. So like they'll bloom with pink flowers, and then eventually at the end of the growing season, they'll be blue. It's not overnight. Don't try to do it overnight. And that's it. So for the Mendelian genetics, you need to know codominance, incomplete dominance, polygenic inheritance. You need to know multiple alleles like blood typing. One that we did not cover is epistatic. And I do want to cover epistatic genes. Okay. If you go to the topics page and you click on the notes from previous years, it's a long slideshow, which is why we didn't assign it to you guys. But for the AP, I do want you to know epistatic genes as well. Okay. So I'm going to kind of get through all of this. If you need a review, it's much more in depth. It's got a lot more text if you find that the review book is not as detailed. If you're kind of curious of like, what in the world is this penguin? That's an icon that I use in my slides usually. If you turn the sound on, it quacks sometimes. It's the only animal sound that they had at the time when I was making them. So if you want more of an example of like law of segregation, if you're not quite understanding like Punnett squares all that much, or you're curious how it would work with a dihybrid cross versus a monohybrid cross, they're on there. Just example, the idea of being familiar with monohybrid crosses, test crosses, dominant, recessive, law of segregation, law of independent assortment, Punnett squares, genomes. Geez, there's a lot of language here. Okay, haploid, diploid, homozygous, heterozygous, loci, locus. These are all terms that we've talked about. They're all terms that you need to know. So it's kind of a self-check, like, where are you? Do you understand what these words are saying? And then I'm going to get down here to my nice Mendelian. This would be like foxes, environmental changes. So when it's cold, their brown fur falls out and turns white. And then when it warms up, the brown fur will pop in. I'm getting there. There we go. Epistasis. Okay. So if you, for coat color, this works really well in mice. It also works really well if you ever seen a chocolate lab. So I'll use this example. So in epistatic genetics, let me make sure I'm sharing my screen. Okay, I'm good. Okay. What epistasis is where one gene, totally separate, okay, this is not the pigment gene, one gene, totally separate, is the on-off switch for a totally different gene for the pigment, okay? So let's talk about the pigment. If you don't have a functioning version of the pigment gene, okay, then you don't make the pigment, okay, non-functioning, okay? If you make a functioning version of the pigment gene, you can have pigment, right? If you, okay, so that's the E. E is the pigment. Where are we? Yeah, dark pigment, no dark pigment in fur. Sorry, that's the epistatic gene. Oh my goodness. E is the epistatic gene. E is the expression. This is the on-off switch. Switch is off, EE. They do not have an on switch for the pigment. Okay, so that's a separate gene. So this is the on-off switch. So their switch is off. Switch is off. At least one dominant copy of the epistatic gene switches on. Okay, so that means that they have the possibility to express the dark pigment in their fur, or they don't have the possibility at all to express the dark pigment in their fur. So here's what you got. Black labs will have at least one dominant pigment gene and epistatic gene. So they will produce the pigment and it's turned on. So yes, they can express that pigment, okay? So they make it and they express it, black labs. If, however, they make the dark pigment, but they don't express it, okay? So they make it, but they don't express it all the way, you're going to be a chocolate lab. Okay. Over here in the yellow labs, they don't have that on off switch. They're recessive. And so they aren't going to express it. Okay. They don't have the on switch. They are not going to express that pigment. If you see a yellow lab that has a black nose, that yellow lab does have the capability of producing the pigment, but its epistatic gene isn't in like a dominant version. They're not allowed to express it. They have it, they can't express it. If they're homozygous recessive for both the epistatic and the pigment gene, then they're going to be yellow with a brown nose. Okay. If they have a black nose, so if you have a yellow lab with a black nose and a yellow lab with a black nose, all the puppies you're ever going to get are going to be probably yellow labs with black noses or yellow labs with brown noses. Okay. Because yellow labs lack that dominant epistatic gene, which means even if they make the pigment, they cannot express it. Okay, that's what an epistatic gene does, is it allows you to express the presence of another gene. Okay, obviously you're not going to express it if you're recessive. You don't have a functioning copy to make the pigment, so you're not going to express the non-functioning copy, right? For chocolate labs, they have the possibility, they're just recessive. And for black labs, they're dominant for both. So here's the cool thing. If you take two black labs and they are heterozygous for both traits, okay, for the pigment trait and for the epistatic trait, they are, this is a dihybrid. These are both hybrids, di meaning two traits, dihybrid cross. These are all the possible allele combinations that the male can pass on in the sperm. because remember the dominant B allele and the dominant E allele, the recessive B allele and the recessive E allele all assort independently from each other. Okay, so these are the 4 different sperm combinations, and these are the 4 different possible egg combinations. And so really we're not doing just a 2 by 2 Punnett square anymore. Instead, with a dihybrid cross, it's a 4 by 4, or it's out of 16 squares. Most of your offspring will look like the parents. You're going to end up with 1, 2, 3, 4, 5, 6, 7, 8, 9, okay, black labs. You're going to end up with 4 yellow labs, and you're going to end up with 3 chocolate labs, or 3 out of 16. You don't end up with 16 puppies. You basically have a 9 out of 16 chance of having it's probability, it's all probability. 9 out of 16 for having another black lab puppy, 4 out of 16 for having a yellow lab, or 3 out of 16 for having a chocolate lab. Okay. So again, if you want to see that with epistasis is where one gene completely masks another gene, or just refuses to allow them to express it. And look at this, like, how would you know that this isn't due to random chance? You do a chi-square test. So on the AP, they could give you a problem with an epistatic gene, give you a block, like a data table block of text with numbers in it, with data of parental genotypes, genotypes of offspring, and then phenotypes. The 9 to 3 to 3 to 1 is phenotypes. And then they could ask you to calculate chi-square. And you would have to know for epistasis, it would end up being a 9 to 3 to 4. And you could then calculate based on those 9 out of 16, 3 out of 16, 4 out of 16. You could then calculate how many should be, or the expected value. Because remember, in chi-square calculations, you need both the observed and the expected. And they're not in percentages, they're actual numbers. So you should put them in decimals. There you go. All right, guys, that is it. Good luck with the notes. If you have any questions, office hours, Monday is a holiday, and I will have my son with me because he does not have class at the JK. But if you have any questions, email me, so I won't be Zooming. Tuesday is a B day. I do have a meeting after school, but it's not till 3:15. And so I will be having office hours from like 2:50 to 3:15 to allow people to ask questions. And then definitely like email me. I will be answering emails on Monday. So if you have any questions, let me know, and enjoy the rest of your weekend and your Monday. And then Tuesday, we will be looking at different types of genetic inheritance and discussing it as a class. All right. Thanks, guys.